keep feeling free to interrupt me if you've got questions or anything, even if it's a little off topic. This is a very focused sort of case study here. We're looking at the effective method of anesthesia on metabolomics of mammalian tissues. But as you've heard today again and again, sample preparation, sample collection are really probably among the biggest factors, aside from designing a good study, the biggest factors in getting good metabolomics data and getting something meaningful out of your experiment. So that's why we're emphasizing it so much. So after this session, hopefully you will be aware of some challenges associated with sample collection for metabolomics, specifically metabolomics of tissue samples like organs from a living animal. Understand some effects of anesthesia and euthanasia on the rodent tissue metabolome. So looking specifically at those of you who may do research with mice and rats. And then hopefully be able to select or determine the parameters that you need to select a tissue collection strategy to help reduce metabolic perturbations. So what you actually measure when you're looking at your sample is reflective of what was happening in the animal and not reflective of just what you did to it to get it out and into the vial. Okay, so here we are, sample collection study design, part of the workflow. Okay. So metabolomics of tissues. First, what makes metabolomics of tissues unique? Why does it deserve special consideration compared to metabolomics of serum or a cultured cell or anything else that you might look at? Then we'll talk about the purpose of the particular data that I'm presenting. This, unlike the other talks, which were very general, was a particular study that we did because we needed to answer a question for our research. We'll look at the methods that were used, so tissue collection methods that I evaluated, the sample and data analysis methods, just to sort of link it back to the chromatography and mass spec that you heard back earlier in the lecture, and then some results. So we'll look at the effect of anesthesia on the metabolome on an untargeted and a targeted level, and then recommendations. What's the best way to do this? And what about human samples? If you ever have an occasion to collect a biopsy sample, what's up with that in terms of metabolomics? Okay, again, just to reiterate, why are we spending so long on sample preparation today? A lot of time will be spent designing your experiment, analyzing the samples, interpreting the data, but you could get absolutely nothing. You could do everything else right, not collect your samples properly, and get nothing useful out of your data. So we don't want that to happen to you. So we found metabolomics of tissue samples to be a particular area where things are most sensitive to perturbation. No sample type is immune, but if you're collecting tissue samples, you can't be naive about this sort of stuff, and so we'll give you our recommendations. Okay, metabolomics of tissues and organs. What makes tissues unique? Biofluids are great. So things like serum and urine are great samples for metabolomics because they represent an average metabolic state of the organism you're trying to study. You can get access in a single blood draw to metabolites that are produced by the liver, skeletal muscle, and any other number of organs that you're trying to study. It's all there in one package. If there is a perturbation, chances are something may show up in the blood. However, it's readily accessible. You can get it from a needle stick. You can draw it from an animal without sacrificing the animal. The biofluids are easy to handle. It's a liquid. You move it around. You freeze it. You thaw it. You can work with it fairly easily. But it's often different. If you find something that's different in serum, where did it come from? Are you looking at a metabolic difference that was originating in the liver? Did it come from a site of an injury? Does it come from, was it a primary effect? If you've got cancer, is it a perturbation due to cancer? Is it due to some secondary effect of some other metabolite that's being produced or some other compound? It's really impossible to say. And also, serum does not cover the entire metabolome. Some metabolites stay predominantly trapped inside the cell. Things that are phosphorylated, nucleotides, nucleosides, these are predominantly intracellular metabolites. And if you don't look there, if you don't look inside the cells, if you don't look in the tissues, you may not see them at all. Okay, so tissues offer a more focused view of the metabolome. They provide direct insights into the sites of metabolic regulation. If you have a disease, it usually may affect an organ. So you're by looking at directly what's going on there, you may get something that's more primary to the disease that you're studying. However, they require more laborious collection and sample handling. In a human, you're dealing with a biopsy, and then you're talking about things that are challenging to do, require a lot more steps, a lot more protocol approval, and a lot more careful attention. In animals, even in animals, it's not too hard to collect tissues, but there's steps you have to anesthetize, usually ultimately euthanize your animal in process of collecting tissue. So what we wanted to do, there are a lot of aspects of sample prep and metabolomics that you've already heard about that have been thoroughly studied. Serum versus plasma. The gist of that answer, as Anna was telling you, both are fine. Just be consistent predominantly. Choice of anticoagulant, there are some that are better than others. Extraction solvents, analytical method, reproducibility, all of these have been studied for cells. Less information when we went, so we wanted to do studies in animals and focus on metabolome primarily in skeletal muscle, but also liver and other tissues. And less information was about the effects of sample preparation on the metabolome. And so most of the literature that discusses sample preparation techniques are focused on things that aren't subject to a rapid turnover. So things like genes, gene expression, protein expression, et cetera. So we wanted to look at 6 commonly used methods of anesthesia and euthanasia, a necessary part of collecting tissues, not the only part of the procedure, and see how they might affect the metabolome, so that we could tell if we chose a particular strategy that the changes that we might observe between our 2 lines of rats that we're studying, or whatever other condition that we were looking at, were really due to the differences that we wanted to, that were from the biology and not from the sample collection. So okay, and so most of the data in this is in this +1 paper that we wrote some time back. There's other sources that are available on this. If you're interested in this specifically, please ask. Okay, so this was an animal study, so everything was ethically approved and done in a manner to minimize use of animals, et cetera. Okay, so here's the overview of the study. So we chose to use C57 Black 6 mice just because they're the most commonly used animal model. We've replicated, or we've done some limited follow-up on this in rats and found that the trends are generally the same. But what we wanted to do was collect 5 different types of tissues which reflect a variety of different tissue types. We didn't look at brain in this study, but we collected skeletal muscle, serum, liver, heart, and adipose tissue, and using 6 different commonly used methods of anesthesia and euthanasia. And then we wanted to look at the samples and look at the metabolome. And the workup of the samples was fairly straightforward. We took our tissues, aside from serum, which you know how we handle that. So we collected the entire organ so as to avoid any sort of site-specific effects. You can, if you collect a heart, the tip of the heart may have a different metabolome profile from some other part of the aorta or whatnot. So we collected the entire organ, froze it, ground it up under liquid nitrogen. This is a relatively manual sample preparation technique, good for relatively low throughput. This study was not too large in sample numbers. If you have a lot of samples, we have other techniques. But then the pulverized tissue samples, while they were still frozen, we add extraction solvent, immediately sonicate to precipitate proteins, deactivate enzymes, extract the metabolites, centrifuge to pellet the residual tissue debris and proteins, collect the supernatant containing the metabolites, analyze it by LC-MS, and then do targeted and untargeted metabolome profiling. Okay, so what are the methods of anesthesia that you study? If you do work with animals, you're probably familiar with most of these methods. So cervical dislocation is kind of it's a method of euthanasia that's conditionally acceptable with mice. You can use it if there's a reason that you have to do it, as opposed to some other mode of euthanasia. It's very fast. It involves no administration of any chemical anesthetic or anything else like that. And it takes about 10 seconds from when you have an animal to when you are able to start collecting tissue. So very fast. This was what we thought might be the preferred method of tissue collection because of the fact that it is so fast and might induce less disruption than other things which are commonly done. So very commonly used, although it's becoming somewhat less, somewhat more controversial in the literature. If you read this sort of literature, CO2 is a very common way to euthanize mice. It takes about 2.5 minutes, and a lot of people will use that as what they do to euthanize their animals before they'll collect tissue for gene expression analysis or for protein analysis for Western blots, et cetera. And so this is very commonly used. You can immediately start to think, CO2 exposure, metabolomics might not be a good combination. But there are those who have submitted samples. There are reports in the literature where this has been used for metabolomics in the past. Isoflurane overdose, so basically using a chemical anesthetic, but still the animals go down, by basically cessation of respiration a little bit faster than carbon dioxide. There's other inhaled anesthetics that you can use. We just chose to evaluate this one. It's the most commonly used in our lab. Another option is to collect tissues while the animals are under anesthesia. So these are still ultimately terminal procedures. We're not talking about survival surgery in this case, but the goal here is to collect tissues, or those tissues which can be collected while the animal is under deep anesthesia, while blood circulation and respiration are still intact. So there's a few options here that we evaluated. One, which is commonly used, is continuously administered isoflurane anesthesia using an inhaled anesthetic machine. Another one is ketamine, sometimes commonly used ketamine xylazine is a common combination, and then another one is pentobarbital. And so these continuous isoflurane takes effect pretty quickly. Ketamine and pentobarbital take a little bit longer to take effect. Following anesthesia, the animals were immediately dissected, surgical tissue collected, and then the samples were pulled out and liquid nitrogen frozen as quickly as we could do, and then stored at -80. Okay, so here are the tissues that we collected, and just some thoughts about them. For muscle, we collected gastroc. That was the first tissue we pulled out. It was done within a minute. Arterial blood was then collected after we opened the abdominal cavity, and that was done with a 25 gauge needle, and it takes maybe 30 seconds to do. Liver, once you're in the abdominal cavity, it's very easy to collect liver. Heart, you then open up the chest and collect the heart. That takes at most another minute. Blood flow is maintained up until this point for anesthetized animals. After you take out the heart, obviously blood flow has ceased. And then we collected epididymal white adipose tissue just because it's the quickest one to get. There's other depots of fat that would be very reasonable to study as well. So these are the tissues we chose to look at. Extraction procedure, as I mentioned before, we cryopulverize, transfer defined mass to the tubes, add extraction solvent. In this case, we used a 7:2:1 mixture of methanol, water, and chloroform. This containing a C13 internal standard mix to help account for differences in recovery that we might get from sample to sample, and to be able to standardize and measure absolute concentrations for the metabolites that we have, the C13 internal standards. And we used a 1 ml solvent to 30 milligrams of tissue ratio. This is just, there's no absolute magic to that. It's something that's worked well in our lab. Probe sonicate for 30 seconds to further disrupt the tissue. It's already been cryopulverized, so it's fairly fine powder, but to really help lyse the cells, release the metabolites quickly, we use this probe sonication. Centrifuge. Then the supernatant goes to LC-MS analysis, and then we dry down the remaining tissue pellet and measure the mass. We try to extract approximately equivalent masses of tissue from sample to sample. Very helpful to get good reproducible results, but you never get it perfect, so the dry tissue mass is a reasonable way to normalize everything out further. Analytical method, just to tie it back again, probably not imminently exciting to most of you, but we used the Hillock approach on a Phenomenex Luna NH2 column. So this, and I'll show you some slides. It worked very well for central carbon metabolism. These are our mobile phases. Our mass spec was done at time of flight, and we were doing full scan mass spec. I feel like I skipped something in there. I guess not. Okay. So untargeted metabolomics. This is the very 30,000 feet level view using principal component analysis of the data. And so if you're not familiar with PCA at all, you will be by the time you're done with this workshop, I guarantee. Basically what this does is it separates groups, or it helps you visualize separation between groups on from all of your data combined into one thing. So into one sort of single plot. All of the metabolites have an impact. Things that separate groups, you may be able to pull out from that. So we did no intervention with these animals, right? These were animals under fasting conditions collected at the same time of day. We did everything else we could to minimize variation. So theoretically, if we'd done this, and there was no difference, we would have gotten the most boring metabolome ever. We should have gotten all of the cells, all of the samples of a given tissue type right on top of one another. Obviously, there's analytical variability, some biological variability, although these are inbred mice, so there shouldn't be too much. But clearly, by looking at these plots, that's not what we got. We got a significant grouping by mode of anesthesia, and it's a little hard to see. We'll look at it, I think, a little bit more in detail in a second. But you can see a clear grouping depending on your tissue, more or less grouping. So skeletal muscle sort of separated out into two groups. Heart, you see, each group kind of looking almost its own thing. Liver, again, two groups, sort of more akin to skeletal muscle. White adipose tissue was a little bit more of a mix-up, so less differentiation between groups. Serum, again, we do see the groupings aren't exactly the same, but we do see some substantial differences between our modes of anesthesia and euthanasia. Okay, so what's going on here? So obviously we knew, as we suspected going into the study, the reason we did it, that there is a dramatic effect of these different procedures on what you measure in your tissue. So we knew we were right to be concerned what's going on. This is just a PLSDA, another way of looking at it. It sort of emphasizes the differences, focuses on maximizing differences. You'll learn tomorrow about the difference between a supervised and an unsupervised technique in this method. The data reduction technique is aware of the groupings, whereas in the other one, there was the computer did not know what the groupings were as it was trying to optimize the PCA, and that's very poorly explained. I apologize for my cringeworthy statistics and stuff like that. So okay, what's going on? Let's look at skeletal muscle. So look at our groupings here. So we have cervical dislocation down here. This was what we thought might be our reference standard best metabolome profile option. Up here we have the cervical. I'm sorry, we have CO2 and isoflurane overdose. So these are methods of euthanasia. The animal was not breathing by the time the tissues were collected. Out here in these three groups, we have ketamine, pentobarbital, and continuous isoflurane. So they overlap with each other reasonably well. You see a little bit, of separation, but by and large they're on top of each other. So we immediately have a distinction between metabolites collected. Even though we did this relatively quickly, we have a big distinction between metabolites, between samples that were collected in euthanized animals versus animals under anesthesia. So this may be one of those, oh my gosh, this is the most obvious thing ever. Of course, that's going to be a big difference. The animals were living versus dead. You'd be surprised how many people say, oh, is it okay if I just sack my animals the way I've always done, collect the tissues and give them to you? And how many reports in literature have not taken this into consideration. In this case, CD is somewhere in between the two and also quite variable. One thing we know about CD is it induces muscle contractions, right? So not only are the animals not breathing when the muscle is finally collected, but also you've induced some pretty strong response of a stimulus, and that can introduce changes in the metabolome. Okay, so just looking at, so that was untargeted metabolomics kind of fed in to the data. We can look at now what are some of the metabolites that drive those changes. So I just did a very basic workup here and looked at the top 5 compounds in each group that were responsible for driving the differentiation from those PLSDA plots. And we come up with sort of a myriad of different metabolites in different tissues. So in skeletal muscle, succinate was one of the main ones. We have some substantial changes in glycolysis intermediates, pentose phosphate pathway in the heart. We saw a change in this fatty acid. We see some purine metabolites, some nucleotide metabolites. Here again, we see some in liver. It was a little bit different. We see some nucleotides. Again, we've got a fatty acid and adipose tissue, various other lipid species. In serum, we have hypoxanthine showing up, xanthine, another nucleotide breakdown product. Some, a pentose phosphate pathway intermediate, which is a little bit unusual, although they are present in serum. So we have a range of different metabolites changed. So then let's look a little bit more specifically in a targeted way. So in terms of the targeted metabolomics, this method that we were using was a sort of a semi-targeted approach. So we're not doing absolute quantitation. We're just doing relative quantitation in 112 known polar metabolites that can be analyzed using the same LC-MS data. We can identify them by known mass and retention time of authentic standards. And we used C13 internal standards to normalize when possible for 21 compounds. Everything else was relative quantitation. And this is just a profile of some of the peaks that we get using this method. Okay, so we can generate a profile of the metabolome and its response to the method of tissue, method of anesthesia or euthanasia in the rodent. You can see clearly, so this is a fold change plot, and obviously you can't read what's going on here. We'll take a look at this in more detail soon. But you can see clearly that so the first one is cervical dislocation. Everything is fold change versus cervical dislocation. And right away you can see that there are pretty substantial disruptions. This is sorted by classes. Here we have glycolysis, TCA cycle, nucleosides, nucleotides. I'm sorry, that's down here. So anyway, difficult to see. So we'll look at these effects individually in the following slide. So here's just a zoom in on part of it. So here we have glycolysis. This is kind of what you saw a little bit of in the untargeted data. But right away you can see the same thing that we observed. Skeletal muscle. We have the three modes of tissue collection where the animals were still breathing, looking reasonably similar one to another. I'm sorry, you have the three modes where the animal was euthanized, looking reasonably similar one to another, and then a big difference with the tissue collection where the animals were still breathing. And so that holds true across tissue type. It's true in heart, it's true in liver. One thing you'll notice that the response is not uniform by tissue type, however. So in terms of glucose and in terms of glucose 6-phosphate, we see elevated levels of hexose phosphates in the euthanized animals, whereas it's the other way around in the skeletal muscle. It's the other way around in heart. So there's some pretty complex effects going on here. Serum, we also see the same sort of effects. The changes are different from what we observe. Not all metabolites are detected in serum, but we see some dramatic effects. So if you're really interested in the specifics of what are going on, I recommend you look at the paper and you look at those details. I'll show you a few things that I think can be interpreted in a meaningful way here. In skeletal muscle, again, I mentioned we see higher levels of glycolytic intermediates in the cervical dislocation, CO2 asphyxiation, isoflurane OD as compared to the modes of anesthesia. What's going on? Well, I think what's going on is that we have increased glycolytic activity when the tissues are exposed to hypoxia, right? So the tissues are not dead. The muscles are not absolutely non-metabolically active. They're just doing what you would expect would happen in the absence of oxygen. They're switching over from oxidative metabolism, more dependent on TCA cycle and electron transport, to modes of generating ATP that are not dependent on oxygen, and that's glycolysis through lactate production. And so we see elevated levels of these glycolytic intermediates. And not surprisingly, we see elevated levels of lactate under those conditions. So that's hypoxia. And that's a general theme about what's going on here. Not the entirety of the data, but a lot of the effects are due to hypoxia. Succinate, another one which changes in a very similar pattern. And succinate also turns out to be a very sensitive metabolite to hypoxia. The mechanism by which it increases is a little bit different, but the data look very similar to glucose 6-phosphate and other glycolytic metabolites. Not everything changes uniformly. Citrate, isocitrate, another TCA cycle intermediate, just like succinate, a few steps upstream are almost the opposite. So we see elevated levels of citrate and isocitrate, perhaps due to altered levels of flux, these things getting consumed down and depleted as the cells shift away from oxidative metabolism would be one possibility. Surprisingly, ATP, you might think, oh, you should be seeing ATP depletion if you're hypoxic. It turns out not. It turns out this is not a long enough period of time. The cells have many mechanisms in place to defend the levels of ATP. Phosphocreatine in skeletal muscle is a big one. So we did see changes in phosphocreatine level as the phosphocreatine was being depleted. If you wait long enough, if we had waited 30 minutes with the animals, post-euthanasia, I guarantee we would have started to see bigger changes here. But a more sensitive metabolite than ATP is AMP. So this is the lower energy state that has not been reconverted in ATP. You do see that in CO2 asphyxiation, isoflurane OD, we start to see an elevated level of AMP. So although the ATP levels are abundant and not changing under this time course, we do have some response of the nucleotides in that case. When you look further downstream, so those elevated levels of AMP, one of the cellular responses to elevated AMP levels is to get rid of the AMP, ideally through resynthesis of ATP, but sometimes that's not possible, and so AMP can be broken down into other nucleosides. And so we saw a lot of evidence of purine metabolite degradation in the liver and also in the serum. We see elevated levels of these breakdown products, hypoxanthine, one of them, guanine, and various other products that were up greater in cervical dislocation as compared to isoflurane anesthesia. So you began to develop a picture of what's going on. It's largely a response to hypoxia. Not entirely, however. Do the anesthetics themselves have any effect on the metabolome? Yes, they do. So for whatever reason, we saw elevated levels of aspartate in isoflurane anesthetized liver samples. And it may be that there's some inhibition in mitochondrial aspartate aminotransferase. That was something that we gleaned from literature, and this may be a specific effect in terms of isoflurane metabolism. The rat's bodies, the mice bodies, trying to to clear the isoflurane, and one of its sites of metabolism is in the liver. We also saw changes that were not associated with hypoxia. We believe that this elevated malate level is sort of closer to the natural state of the animals. Ketamine, on the other hand, showed depleted levels of malate, more akin to what you saw in cervical dislocation and CO2 asphyxiation. So it's not as simple as saying, breathing good, not breathing bad. That's the first approximation. But there are other more subtle effects that you should potentially be aware of. Okay, so what does this all mean? If you're going to collect tissue, what should you do? So I recommend, and I think others are coming on board with this, that tissues be collected under anesthesia for the purposes of metabolomic, with blood circulation intact, and because this shows less evidence of hypoxia. We recommend, as far as if you're trying to study a phenomenon that is time sensitive, continuous isoflurane anesthesia, because it is fast to induce and allows fast initiation of tissue collection. Of course, it does have its effects. We just saw in liver there was that elevation of aspartate. There weren't that many other changes, but you should be aware of potential effects of any anesthetic or technique that you're using. Regardless of the mode of anesthesia you use, very important to document what you do in publications. I can't tell you how many procedures come out in literature where there's no mention of mode of anesthesia, no way to tell aside from writing the author how they did it. And so I would urge you, if you're doing this type of work, to report that, even if you have to do something that's not quite ideal for it, because you need it for another part of your procedure. At least you've said what's been done so that someone else can reproduce your results, and that they're aware of what sort of effects might be there. Of course, you have to follow all guidelines for ethical treatment of animals, and if possible, we'll reemphasize, consult with the core lab first before you do your studies. Be it an animal experiment or an experiment in cells, it's worth doing. Okay, so just a brief word about what about human samples? Is there anything we can learn about what you should do if you're collecting human biopsy samples for those of you that are involved in clinical research? Obviously, we don't have as much flexibility in collecting biopsy samples in people as we do in animals. The sampling method is constrained. You're only going to be doing anesthesia. You're not going to be euthanizing your human subjects to collect their tissues. Hypoxia is less likely to be a problem because you're using anesthesia. However, speed of tissue collection is still a concern. do you want to spend a long time rinsing your samples, getting them carefully dissected, free of all debris? If it's essential to some other metric in your study, maybe you have to, but all other things being equal for purposes of metabolomics, you want to do it fast. You want to immediately submerge your samples in liquid nitrogen, minimal rinsing steps. You must also consider the presence of anesthetic and its effects on the metabolome. If you do a lidocaine anesthesia, that lidocaine, you better believe, is going to show up in your muscle. It'll be the most abundant peak. And so be aware that it's going to be there. I can tell you stories about discovering that on a couple of occasions that will affect your interpretation of the data. And again, consult with the core. Okay, very good. So thanks for the people who helped me with this particular study, and happy to answer questions if you have, if we've got some time. Sure. Yep. Yep. Good. Very good question. Yeah, right. Totally, very good, reasonable question. So there's two types of degradation that can occur to a metabolite. One is enzymatic degradation, right, due to other biological enzymes remaining active in your sample, and that's the biggest problem, right? So when you have a tissue sample, if it's when it's been removed from wherever you're studying it, you leave it at room temperature, enzymes are still active and they're turning over metabolites and they're responding to the situation where they're in. If you take a tissue sample that's been in the -80 and you thaw it and you don't do anything else to it, those enzymes come back to activity and they're going to wreak havoc on your metabolome. The key thing that happens between then is we add an organic solvent, right? And then we disrupt the tissue. So immediately upon freezing, the very next thing that happens to your sample is that solvent goes on there and we break it apart as quickly as we can. That denatures enzymes, right? So it should remove that enzymatic activity ideally instantaneously. And so you've lost that concern about the enzymes wreaking havoc on your metabolome. What's left is chemical degradation, right? So the things that can happen to a metabolite sitting in solution as it may break down, and so that is a concern. There are metabolites that are labial, even in free solution with no enzymes present, but they're many fewer than what could be affected by enzymes. So if they are an issue, if you're trying to study, for example, succinyl-CoA, then I would recommend strongly against a drying procedure in your metabolomics unless you have some way of chemically stabilizing, because that's a metabolite that will chemically degrade. For most metabolites, after you've denatured your proteins, the metabolome is stable. So it is a balancing act. You do have to consider it, but the key thing is that deactivation of your enzymes, and that allows those things like drying steps to be at least workable for most of the metabolome.
So when you add a solvent into your samples, do you control the temperature?
Yes, so we do the best we can. So I like to start with sample with extraction solvent that is chilled to 0 degrees or -20. You start getting to -80, stored on dry ice. You have challenges with pipetting, and you may not effectively disrupt your sample because it stays frozen for so long in solution that it takes a while to break apart, and that's not ideal. But yes, we like to control things. Sonication, you can induce heating of your sample, so we like to keep the sample maintained on ice while we're sonicating to the extent possible. So yes, cold is good, and we try to keep it as controlled as we can.
I wondered if you thought samples that have been extracted and then frozen in the freezer for a very long time.
Can you go back to them?
Okay.
It depends on what you're looking for. If you're looking for a metabolite that's chemically labial, then perhaps not. The amount, you can definitely see, oxidation if you're looking for, or if you're looking at oxidized lipids or things like that, those can chemically degrade by just, interacting with oxygen in the atmosphere, and you can lose things. Most metabolites, if you have a well-sealed vial in an organic solvent, are reasonably stable. So again, the ideal is to, do everything quickly and, analyze your samples fresh. A lot of the I know this is a hand-waving kind of an answer. You'd need a good thorough study in your particular type of sample to really answer it, and not too many of those have been done. But there are metabolites that are just fine under those circumstances, and you can go back and run a metabolome, and the profile looks the same as it does as when you started. So. Dry, it's better. Dry is usually better than in solvent. So we will often, if we know we were going to want to go back to samples later, you may dry down and then put them in the freezer and leave them sitting there. Again, things that are sensitive to oxidation in the air may be unstable even under those conditions. But yeah, generally dry is better than insolvent. Cold is better than warm. Surprise, surprise. Yeah. One last question. Yeah. Right. Right. So in general, I will say, and this may or may not be answering your question, but in general, surprisingly enough, in spite of disruptions that are induced by method of anesthesia, you often can still pick out the biological signal on top of it, right? So if you did a consistent method of cervical dislocation, you'd obviously be dealing with a good bit more noise there. You saw the variability in the metabolome, but you may still be able to detect your biological effect. You may find, yeah, if you had a 1.5-fold change from isoflurane anesthesia, and you had a 3-fold change due to your disruption, probably that disruption in the metabolome due to what you actually wanted to study would still be there. Again, it's a case-by-case, metabolite-by-metabolite sort of basis. Things may be affected more profoundly. Some things may be completely depleted, and then you've lost all your signal. Some things, even though they're notably disrupted, you may still be just fine in detecting a change. So whatever you do, report it and do it consistently. But there are best practices and then less ideal procedures among those choices.